And they can do anything. Okay, so evolution pushing the boundaries of catalysis. So this is a protein encoded by a bacterium that wants to obtain nutrients from leaf. So this is Xanthomonas, it's a plant pathogen, wants to get nutrients from leaf. The easiest way, and easy is the wrong word here, the easiest way for it to do this is to encode for a protein that can cause ice crystals to form. And the ice crystals form in the veins of the leaf, and they rupture it, and then the bacterium can take the sugars. And when you see it in action, right, so they've got cold water, super cooled water in there, and they're going to add this protein using the dropper. So you can see it kind of diffusing.
How's it going?
You know it's not going to work this time, don't you?
Of course.
And they're adding more and more now.
And
Really?
that's so disappointing.
Oh my gosh.
One more. Oh, there it goes. There it goes.
There it goes.
Is that not awesome?
That is
I'm
beautiful.
going to back off. I want you to turn it upside down. The top's on it, right? Go.
Right.
That
That's
one's
amazing.
a good one.
That's absolutely amazing. So proteins can pretty much do anything, right? Irrelevant of what else is going on there. So proteins can do anything. Evolution put them to do anything, right? And if you were to look at that protein, you're like, how on earth does it cause ice crystals to form? You'd look on its surface and you'd see it has hydroxyl-containing residues that have exactly the same spacing as crystalline ice. So in other words, it provides a nucleation point that looks like ice, and then it's entropically favorable for the waters to bind and make crystalline ice. And you know, you'd never get anywhere near that without the ability to purify, manipulate, and investigate the protein. And purification is everything, because you need, if you've got a big dirty mix of proteins, you're not going to get clean function out of it. So you need a clean protein, you need to be able to selectively enrich for it in solution. Okay, so I mean, here's other examples of where proteins can be used to some pretty amazing phenotypes. So on the left, in the middle, so this is an electron density map, you can see a buckyball, which is one of those amazing self-contained spheres of carbon that are completely artificial. And what they've done here, they've simply selected for a protein that can bind this. And it's absolutely amazing. It traps this buckyball within the lattice, and you can see it, and the density for it's unequivocal. And so, as you'd know from antibody work, you can pretty much engineer any protein to recognize anything, even if it's not natural, like this buckyball. And then, for example, on the right-hand side here, we have an environmental isolate from an acid mine in South Africa. Why is that interesting? It's interesting because it contains one of the few examples in nature of a cell that's a completely weird shape. You've got these star-shaped bacteria. And of course, this is encoded by a single protein present at the surface that allows them to be star-shaped. Why are they star-shaped? We don't know. But it clearly confers them an advantage. Right, so our aim, and this is monitored here by SDS PageGel, our aim pretty much is to move from the left-hand side to the right-hand side, and you can see in stage 1, got a humongous number of bands there, and our protein of choice, which is mapping to 40K. So these slides will be up, so you don't need to copy too much. You just kind of need to follow me talking. Our protein of choice, which is 40K there, represents, I would say, about 30 to 40% of the cellular material. And over time, we've got these different stages of purification. 2, look, now you get rid of this major contaminant, which was equal amounts with the protein of choice. 3, you enrich again. 4 is probably your most successful step here. And 5, you end up with a pure protein there. So the aim is always to enrich and to minimize loss, really. Loss is the big one. So until you come to purify a protein yourself, you don't quite realize how much biological materials stick to what you have in the lab. So glassware, plasticware, unless you buy really expensive reagents, proteins, DNA, just stick to everything. And your biggest enemy is loss. So proteins are produced in the context of the proteome. Each member will have unique characteristics. So they're the obvious ones, right? Size, charge, shape, post-translational modification. And then it's your job at this process to take selective advantage of that. So when you're doing the enrichment, you'll do one step based on charge, and the next step you do will be based on a different thing. So your next step will be based on size, etc. So to purify it, you should always pretty much understand the physical properties of the thing you're purifying. And quite often, right, that is how stable is it? How stable is it in the conditions of what I'm trying to purify it in? Can I increase its stability? Or can I optimize its stability at least? This new generation of protein-based drugs is massive. So you'll notice quite a lot of the new drugs licensed are pretty much antibodies. And the trade names usually have an I and a B on the end. And they're antibodies raced against epitopes for things like cancer. I think the big examples there are Herceptin, and even things like sepsis, right? Sepsis caused by Staph aureus in the blood. One of the major drugs you can use is an antibody that recognizes the fragments that Staph aureus is releasing to activate the immune system. So things as disparate as a bacterial infection and cancer can both be treated by protein-based drugs. So I mentioned Herceptin there. And I think, right, especially biotechnology, as we get better at learning the principles behind a protein recognizing its target, a lot of our drugs are going to be protein-based drugs rather than small molecules. And of course, if you're injecting them into a patient, the purity's got to be massive, right? It's got to be massively high. You simply can't inject a mix of things into patients. That's not going to work. So you need to consider all these things when you're starting out, when you design your protocol. So objective number one, right? So you know what you want, and it depends on what you're going to use your purified protein for. So purity, right? So where am I going to end up? How pure do I want it? How much effort am I going to put in? So moderate, this can be lower than this, right? 95% here is the upper end there. You can be dirtier than that. So for instance, when I say I want an antigen for antibody production, what I'm doing there is I'm making a protein, probably recombinantly, and the aim is then to either inject it into rabbits, goats, sheep, bleed them out, and then purify the antibody against that protein. And that's a nice way of doing it, right? If you do antibody generation by injecting the pure protein, that's a lot nicer to the animal than the alternative. So what you could do, and I don't like this, is you could run a gel of your protein. Even if it's a big mix, you cut the band out. And then you've got it in the gel, and then you inject that into the animal. Well, that's not very nice to the animal. So antibody generation is better with more purified proteins, and this is a pretty high-end example of how pure you would need that there. N-terminal sequencing as well. You do need moderate. Where would that be useful, right? Where would N-terminal sequencing be useful? It would be when you have an activity that's related to a protein and you need to know the identity of it and mass spec is failing. So you then terminally sequence it. If you've got a really pure sample, your reads are going to be good. You could probably read 15 amino acids, and then you could work out what protein was giving you that activity. So N-terminal sequencing is from the older days of biochemistry, but it would still be informative. Or another example, right? So someone in my lab has just purified a protease, and it is co-purified with its substrate. And it would be informative for us to do N-terminal sequencing, because then we get two reads. We'd get one read for our protease, we'd get another read for our substrate, and we could work out what type of proteins our protease turns over. So it almost gives you information then in like a functional sense. Okay, so you need moderate purity for that, 95%. Then we get into the more biophysical characterization. So if you want structural studies, NMR crystallography, you need to get up to the high. So the higher the better. So 95% here wouldn't be so good. 99 is more what you're aiming for. Cryo-EM, you can kind of select the particles out. You'd still prefer them to be really pure, but cryo-EM allows you to be selective. So the more modern structural techniques, you could get away with being at the lower end there. And then at the really high end, 99%, is where the pharmaceutical companies developing protein-based drugs need to be. So they need to be essentially homogeneous, just that protein. The other reason a company would need it so pure is because if you're making it recombinantly, a lot of the other material present would be immunogenic. So, you know, you want to purify it away from all the cell wall, all the membrane lipids, because otherwise, if you inject that into a patient, their immune system is going to react to it badly. So right at the top end there, you need massive degree of purity, 99% to 100%. So these are some gels, just from my lab. What you want to appreciate here is we got 3 targets, 1, 2, and 3. And what I want to show you is when you when you start out with more protein being made, it makes your job easier. So the higher the amount of protein present initially, the more it suppresses all the contaminants. So that makes sense, right? So if the cell is fully committed to making as much of your protein as possible, it will make less of the contaminating proteins, and it will be easier for you to purify it. So for example, in example number 2, look, my protein's the dominant protein. I can purify it almost straight away. You got a little feather here. Not really sure what that is, but it's essentially pure. Whereas with example number 3, I'm less dominant. That band's starting out in the raw cell lysate. That band's less dominant, and I have a harder time of purifying it, basically. Number 1, that's really pure. And even then, look, you get faint traces of other proteins present. So they could have the same characteristics as your protein, so you're not purifying them away, or they could stick to your protein. And what you should be doing is running those purification steps with an increased amount of salt so that it doesn't stick to it anymore. Now, amount is our second objective, right? So when you work out, I'm going to need it this pure, you can then correlate that with I'm going to need a certain amount. And that's going to define how much you have to start out with. So like the real olden days of biochemistry, you only worked on stuff you could get massive amounts of, which is why the first enzyme ever really investigated was lysozyme, because you could purify it from egg white. And you could just, you know, take a bunch of eggs, off you go. Or, for example, one of the first structures solved was myoglobin from sperm whale. And if you bleed a whale, you get quite a lot of material. So the olden days of biochemistry used to work on things you could have a lot of. Now that we've got very successful recombinant systems, you can take the gene, put it into a different host, make a large amount of it. And that kind of changes things. So, depending on the amount you want, Again, coming back to N-terminal sequencing, you need somewhere between 0.1 and 1 mgs. Raising an antibody, depending on the antibody generation procedure, how many animals you would inject, how much antibody you want at the end, that would determine how much you start with there. For structural studies, it helps if you have a high number of mgs. And then coming back to therapeutics that are going to be made by companies, it's going to do them no good unless they're able to get kilo amounts of a protein to bring to markets, which is why, you see so many fermentation setups to produce huge amounts of therapeutic protein. So let's see what our starting material allows us to do. Plant, animal, microbial. So most of you working with proteins in your projects will probably be using E. coli, and I'll gear that to this. But you could also be using tobacco as a plant system, animal system. You could be using Chinese hamster ovary cells, or even fungal expression. You could be using Piscia if you want glycosylation on your protein. But most of you will be using something as simple as E. coli. Extra or intracellular. So what I will say, right, is extracellular proteins tend to be more stable because they have to survive in a less supportive environment. So the cytoplasm's pretty stabilizing, right? But as soon as you leave the cytoplasm, you have to have disulfide bonds, you have to fold better. So extracellular protein tends to be more stable. Recombinant or natural, I've just spoken about. Like, are we going to take a gene from our system of choice, introduce it into E. coli or another expression strain, or are we going to start with the whole material from our target? So, like an example here would be the researcher who used to work next door with me used to work on membrane proteins from cows. So we would go down to the abattoir, get a beef heart, grind it up, and then isolate the protein from there. So you don't always have to be recombinant. And sometimes, in the natural system, your protein, right, has got other factors that help it be active. And you're only going to get that from a non-recombinant system. So don't feel as though you always have to introduce your gene into an expression strain. Abundance. So you can influence the abundance before you even start purifying. So if it's recombinant, you would run a bunch of tests. You would take your E. coli, and you would say, how much of my protein do you make in minimal media, like M9? How much of my protein do you make in rich media? How much do you make at 37 degrees, which is the optimal growth temperature for coli? How much do you make at 15 degrees? And you can shock expression strains. So if you take E. coli that should grow at 37 and you grow it for half an hour at 42, it goes into shock. It makes a bunch of chaperones, and those chaperones help fold your protein. So the protocol at which you grow the cells can massively affect the abundance of the material you start with. And that should always be a variable in your experiments. So these factors all affect us starting when we start our purification. But for me, right, it's not like I have a massive amount of money as a researcher, but I care less about cost. I care more about quality because I'm looking to use that protein in an experiment. If I worked for a pharmaceutical company, I would be more concerned about speed and cost because that's how my therapeutic drug is going to be judged on its activity per weight, on the cost it took me to make it, on how quick I can make it, and the man hours that go into that. Okay, so industrial use and academic use are going to want different ways through this pathway of purification. So here's our steps. Now, usually, right, this is presented as a very linear flow scheme. it's not always the case, right? I just told you earlier, you can lose your sample quite easily. It sticks to things. Maybe it aggregates over time. Maybe you've got to be quick because your protein is unstable, right? If your protein is unstable, then you might have to commit to doing it all in a day. So on the left-hand side, we have purity. On the right-hand side, we have number of steps. So we would start off, before this even, by optimizing our system, our cellular system to make the protein. We would then prepare those cells for extraction. We would clarify it, so it's obviously going to have a lot of gunk when we break the cells open. So we would clarify it, usually through centrifugation. Then we've got to capture our protein of choice, isolate it from contaminants, concentrate it to a small volume, high concentration, and essentially stabilize it. Then polishing, we get the final stages up here, where if we really want 95 to 100% pure, we're looking at like, okay, now I've got to get rid of that contaminant. Now I've got to get rid of that contaminant. And I'm achieving high purity, but maybe my yield is decreasing because the greater the number of steps here, the lower the yield at the end. So these would be the things that influence us. Okay, so you need to develop an assay, and that's going to influence you, right? Are you looking for a function? So are you following the purification of an enzyme? If so, the assay is obviously your enzyme assay. But quite often you'll be purifying a protein that isn't an enzyme. So what's your assay then, right? So you need a means of determining how much of your selective protein is in there, what its activity is. I've mentioned here minimize the use of additives. I'll come back to that later. So the more things you add in, well, then you've also got to consider that they're kind of contaminating your solution too. You tend to remove damaging contaminants early. So if your first step removes proteases, then that'll help you. You don't want proteases co-purifying with your protein and degrading it over time. And so the first consideration there, right, is you probably make your protein in a cell line that has certain proteases removed. So like a knockout strain of proteases. The trouble you have there is what if those proteases are there for quality control purposes? So when you knock them out, you can now make an unfolded protein and the cell doesn't care. But if you have them in there, the cell does care, and all the protein you make will be active. So as soon as you knock out those quality control proteases thinking you'll get a better yield, well, you will, but it won't be as well folded and as active. So you have to keep that in consideration too. So this is a theoretical chart, but it pretty much replicates what you see in the lab. So if you start off with 100% the amount of your protein you started with, and you say, I'm going to do X amount of purification steps, this is just simple maths, right? If I'm 80% yield every step, by the time I've done 4 steps in a row, I'm under half the amount. By the time I've done 10 steps in a row, I'm down to 20%. And 80% is a ridiculous overestimate at how good you will be. 50 might even be a ridiculous overestimate at how good if you lose 50% each time, it's like a half-life. But by the time you've done 5 steps, you've essentially got nothing left. So you do have to keep that into consideration when you talk about number of steps and the time it takes you to do this. So step 1, recovery of our protein from the raw material. Most commonly microbial, and the reason for that, right, is E. coli is your workhorse. It's cheap. It's well characterized. You know, you could go to pretty much any one of 20 labs here in the building. They'd have different flavors of that. You know, you'd have one E. coli that's specialized for making membrane proteins. You'd have one E. coli that's specialized for making certain types of post-translational modification. So most commonly you go down that route. And then if you have chosen that, if you have chosen to make your protein in a bug, the three ways you would start cracking it open is you'd either use sonication, you'd use a French press, or in a kind of harsh treatment, you'd use chemicals to break the cell open. So we'll go through those now in a minute. Tissues, right, so remember I was mentioning you might be the guy who goes down to the abattoir, takes the beef heart, and decides you want to work on a protein from beef heart. Well, what are your options there? The polytron, which is essentially a blender, will break those tissues up into cells. Or, and this is the way my friend used to do it, this is quite good, pestle and mortar. So imagine you've got raw tissue, you drop it into liquid nitrogen, you essentially vitrify it, and then you bring it out, put it into a pestle and mortar and grind it. You've reduced it to a dust, basically. You've been able to break that tissue into its component parts. So that sounds pretty low-tech, pestle and mortar, but it's actually quite effective. Okay, this is one of my proteins pages to tell you proteins can do anything. This is it's an amazing protein called encapsulin, and you can see one copy of it there in red. Then you can see it as this centrosymmetric symmetry to generate this 1, 2, 3, 4, 5 pentameric kind of patch, and then you can polymerize that to make a shell. So out of one protein, you've made this nano compartment, and then inside the cell, you'd have another protein inside there performing a chemical reaction that is otherwise toxic to the cell. So this is why it's called encapsulin. You can encapsulate that activity and not poison the cell. Well, that's you know, it's almost right. You could almost argue that that's almost like a virus capsid inside the host cell. Beautiful. Right, sonication. So sonication is good in the way that it is cheap and easy. And you probably be the thing you have greatest access to in any biochemistry department. So you go in, you'd have a set of ear protectors on. And if you're good like me, you'd be doing this in a small room, and you'd place a set of ear protectors on the door handle so that anyone who comes into the room doesn't get a blast of sound waves into their ear. And so you protect your own hearing, and you break the bacterial cell wall by using these high-frequency sound waves. So in other words, you've got a tip that vibrates, generates sound waves in the solution. Those result in cavitation, so you get these kind of bubbles that then collapse, and then the pressure of those bubbles collapsing breaks the cells open. You kind of tear the cells down by inducing cavitation in solution. And the only problem you have with this is that it generates heat. So the whole process generates heat, and you don't want to cook your protein. So around the outside of this container, look, you've got a large amount of solution here. You can, in sonication, you can crack quite big volumes of cell paste. Your tip sits in it, usually sits about an inch deep in the solution, and your container will sit in a bath of ice. And you'll do a burst of sonication, and you'll wait, and you'll let the solution cool down again. Then you'll come back, you'll do another burst. And dependent on how old your sonicator is and how well you've set it up, and tuned it, you'll require about 7 or 8 bursts. And your efficiency is not great, right? You'll still only lyse about 70–80% of the cells. So it's cheap, it's fast, but it's not massively efficient. And it can denature the protein if you're not lucky. So if you're working on, let's say, a membrane protein complex, you probably wouldn't choose sonication. This is the thing you would choose if you're working on a membrane protein complex or a sensitive protein, or if you want massive efficiency. This is a French press. And this is a really nice method of lysing your cells. So you have a very, very heavy, very nicely machined chamber here. And if you pick this up, the one thing you don't want to do is drop this on your foot. So I've seen this break someone's toe. So you've got a heavy, heavy chamber. It's beautifully machined, so you have to treat it very carefully. And in the middle, there's a chamber. And into that chamber fits this T-bar that you can see here. And on the end of that T-bar is a rubber seal, an O-ring, that allows it to seal off the top of the chamber you can't see inside. So you plug the top with that T-bar. And on the bottom, you've got another T-bar that screws in. And dependent on how hard you twist that, you can seal off the chamber at the bottom as well. So now you've got your cells inside here. This you've pre-cooled again, right? By the way, you don't heat to be your enemy here. So you'll have pre-cooled this in a polystyrene box full of ice. You load your cells in, and this only usually takes about 30 mils of cells. And you don't want to be lazy, right? You don't want to be lazy and stick all your cells into 30 mils and load in a big gloopy paste. You might want to do 2 runs, right? You might want to do 2 runs of this. You put your cells in, and the container then fits in this ram, and then you discover why this is so heavy and beautifully machined. The hydraulic ram brings this down at the point at which it hits the T-bar, and then it creates a pressure inside that chamber, and then you relieve the pressure by unscrewing at the bottom, but you maintain just the amount of pressure needed to fracture the cells. So as you compress the cells and they come out the bottom, they shear and they lyse open, and your efficiency is amazing, right? Because these have only got one way to go, these cells. Your efficiency of shearing and lysing the cells is beautiful. And they come out the bottom, and you might put them in again. You might give them 2 runs through the machine if you're really desperate, but usually 1 run's enough. What you don't want to do is give this apparatus to someone who doesn't know what they're doing, because then they'll misalign it, and it'll come down, and it'll buckle. You know, it operates at thousands of pounds of force. So you have to align this really nicely. You have to treat it with respect. And the cells come out the bottom, and then you'll get really good efficiency of lysis. Chemical disruption. You might do this. Maybe you do this on a small scale, right? Let's say you've done 10 little flasks of different growth conditions for your cells, and I want to check which one of those 10 on a small scale level gives me the most protein. So instead of sonicating all 10, or even French pressing all 10, what if I just add a chemical reagent that will lyse the cells, and then I can run my samples out on a gel, and on a small scale work out which one's the best. So these are commercial preparations. They contain secret ingredients, but I can tell you now it's usually an enzyme to digest the cell wall, and it's usually a combination of detergents to disrupt the membrane. So those 2 agents together will lyse the cells, but it's pretty nasty way of doing it, really. And the efficiency also is not great. And you'll see this. This is usually labeled as something like bug buster, or you can get different flavors for different expressions. So B per is bacterial permealization, Y per is yeast. So they obviously need different enzymes. If you're breaking open yeast, you'd benefit from a chitinase, not a lysozyme. As I said, I'll put these slides up in full so you can look in your own time. So now we need to remove bulk impurities, right? We've cracked our cells open. What are bulk impurities? So like, you could imagine all the things present in a cell that you really don't want present in your final samples. You've got DNA, RNA, cell wall fragments, organelle fragments if you use the eukaryotic system, other proteins, lipids. So if you want, you could precipitate most of those out with ammonium sulfate or polyethylene glycol. Hope your protein isn't attached to them, right? What's to say your protein doesn't localize to one of those things? So you should be very careful during the precipitation step. Heat treatment is a brilliant one, right? So let's say you were interested in a protein function that can be found in any cell. And you'd be like, I don't care what organism I choose this from. You know, I could get it from human cell, I could get it from a plant, I could get it from a bacterium. But what I'd recommend is you get it from a thermophile. So like, as of about 20 years ago, it's quite the thing to pick a thermophile that lives, so something like Methanococcus lives at 90 degrees C. All of its proteins will be stable at 90 degrees C. You clone it into E. coli, and you can heat E. coli up to 90 degrees. Everything will precipitate, and what's left in solution is your protein from your thermophilic organism. And if you do this, you can even use the PCR machine. So I used to do this quite a lot. You clone the thermophilic protein out. You set the PCR machine to ramp the temperatures up. You know, you can have tube 1 to ramp up to 81, tube 2, 83, 2 degrees steps, and you can work out the precise temperature at which your protein stays in solution, but everything else from your expression strain pellets. So it's quite nice to be able to use a PCR machine to purify proteins. pH treatment, right? You could have a protein from an organism that's present at the extremes of pH. So that star-shaped bacterium earlier, right, lives at pH 2. And you would imagine most proteins in E. coli aren't stable at pH 2. So you could acidify the solution, you could alkify the solution. DNase and RNase treatments can be important, right? So when you crack the cell open, if you did sonication, those sound waves would shear the DNA massively. So sometimes in other applications like genomics, and you want to shear DNA, you'd use sonication. But for something like the French press, by the time your lysate comes out, it'd be very stringy because the strands of DNA haven't been broken. And the viscosity of DNA is amazing. If you've ever seen a solution of DNA, it just pulls the liquid with it. So in order to make your sample malleable and usable, you probably add a few little grains of DNase. RNase, right? RNase is everywhere. So if any of you work on RNA, you'll know this, right? RNase is just everywhere. So maybe you don't need to use RNases. DNases are more important. So you want to prevent degradation. If you keep your samples at 4 degrees, the only real reason you're doing that is because the proteases have their maximal activity at the growth rate of the organism. So like, for example, proteases from E. coli are very good at working at 37 degrees or room temperature, but they're rubbish at working at 4 degrees. So the reason why you're keeping your samples on ice is to lower the activity of those contaminating proteases. So keeping things cold is good. unless, of course, you've got a protein that precipitates in the cold. And there's enough of those. Minimize the time scale, as I've mentioned. Protease inhibitors, so like PMSF will inhibit serine proteases. You've got cysteine protease inhibitors, EDTA, as long as you're not using a nickel resin. And sometimes you can get these combined in a cocktail tablet. But what you should note, right, is that most of the things that give your skin its suppleness are proteases. So don't splash protease inhibitors on your skin. If you're handling protease inhibitors, always use gloves. Otherwise, you'll get pretty nasty looking skin after it gets on you. Oh, sorry. And as I mentioned, they usually come in a small tablet as a cocktail of different inhibitors. You could make the argument that if you need a protease inhibitor tablet to keep your sample active, then your sample's not sufficiently stable anyway. You can sometimes argue that. Some more cool proteins. What do these two proteins do? I love these. These are proteins that have evolved to mimic DNA. So how cool is that? So here's one, a fluoroquinolone resistance protein. It twists. It has the same curvature as DNA. And you put charges on it, and it has the charges in the right space. It even has something that mimics the groove of DNA. And then this protein will dissociate DNA from its target because it mimics it so well. Same for this protein on the left. Sterically, they've evolved to mimic DNA, which is amazing. Right, so you should know something about the protein you're using. Whoops. Hang on a minute, what have I done there? So you can predict, you can't always feel that your prediction is going to be 100% correct, but you can pretty much predict charge, size. Hydrophobicity is a difficult one to predict. Ligand binding, if you know the role of your protein, you usually can't predict stability or solubility. What you should do, first protocol, as long as you're not working on an organism like mine that looks like nothing else, you should check the literature for something similar and see how they handled it, see what they needed to stabilize their sample. And then something like prop-param will tell you a bit statistically about the sequence of your protein. Predicted can be way off though, right? Imagine you've got a protein that's phosphorylated. That's going to carry much more negative charge than the raw sequencing proteins. So you paste it into the box. It tells you the predicted isoelectric point, which will come important now for ion exchange. Then first thing I look at is the number of cysteines. Right? If the number of cysteines is even, and I know my protein is extracellular, periplasmic, whichever, then I'll think they probably form disulfides. And then the first thing I'll do is I'll do sequence alignment with homologs. I'll say, are those cysteines conserved? And if they are, then I will probably use a strain that has had the thioredoxin or something knocked out so it can make disulfide bonds easily. So you should always, first thing you do when you see the sequence, you should always count the number of cysteines. If it's an odd number, then you can pretty much guarantee that one of your cysteines is free, and you should add a reducing agent to stop your cysteines meeting each other in solution and just making chains of your protein. So cheapest reducing agent is something like beta-mercaptoethanol, but it's volatile. More expensive ones like TSEP are better at reducing and staying in solution and keeping your protein happy. Next thing I check is number of tryptophans, because that's going to be the dominant thing that gives you an absorbance at 280 nanometers. You can follow your protein. So let's say you're working on a protein with 0 tryptophans. When you put it in the spectrophotometer, it's going to tell you you've got no protein, when in reality you've got lots. So first thing, count the cysteine. Second thing, count the tryptophans. Tyrosine and phenylalanine do contribute to your A280 signal, but they're pretty minimal. And then that will predict an extinction coefficient, and that will tell you, when you put it in the spectrophotometer, an absorbance of this amount means you've got this much mgs per mil of protein. So it's a conversion between the number of tryptophans, tyrosines, and phenylalanines, and the expected signal if it was just your protein alone, with no contribution from any other protein. And again, that's an estimate, right? That's just an estimate from statistics. That's not the actuality of what you'll see. So off we go with our recombinant protein purification. We might have precipitated out some of the DNA and some other proteins first. Ion exchange might be one of our first ports of call if we're not tagging. So like, here's an example of the protein fold of something that's ribbon-based, and then we put charge on, and you can see negative charge is in red, positive charge is in blue. So when you have a predicted isoelectric point for your protein, that tells you the pH at which your protein has no net charge, but that net charge, right, just means summing up all the negatives and the positives, they cancel each other out. But you've still got patches. So the predicted isoelectric point might not be representative of the ability of your protein to bind the resin anyway, because you've got selective patches that could still bind. So the PI responds, the isoelectric point responds to the global charge, but you've got local charge in pockets. And I've mentioned those complications you get via post-translational modification. So the big one, if your protein is heavily phosphorylated, it's obviously going to be more negatively charged. If it's methylated on the arginines, it takes away the positive charge. You know, other things can affect the charge by being modified. Then what you want is to have a resin that has the opposite charge to your predicted protein. So if your resin has a strong positive charge, it's probably a quaternary amine. So this is something like Q-cephorose. And then it will bind your protein that has a negative charge on it. DEAE has a less strong positive charge, so it has a weaker affinity to your protein in terms of the Q does. So you can use these two resins differentially. Likewise, if you've got the opposite charge and your protein is positive, it's going to stick to a negative resin. And a strong negative resin is SP, this sulfur propyl group, and a weak negative resin is this carboxymethyl group. So you work out what the expected charge on your protein is, then you pick one of these four, usually. So let's say your isoelectric point is predicted to be 957. If you're under that, then your protein is going to be positively charged. And when I say under that, I would say always 1 pH unit away from the PI. I wouldn't approach, I'd be at least at something like 7.5, 8 there. I wouldn't approach the isoelectric point in terms of pH. The other issue you have, right? Let's say your protein had an isoelectric point of 957 and you're at pH 9.5. Then your protein has no net charge. It might fall out of solution. So charge is one of the things that keeps protein in solution. So usually, not always, as you approach the isoelectric point in terms of pH, your protein will just precipitate. So usually you want to know an estimate of your isoelectric point, and you want to keep the pH at least 1 unit away from that. So if we follow this case, we would pick a resin that has a complementary charge to our protein. So at pH 7.5, we'd have a negatively charged resin. If we wanted to bind our protein weakly, we'd use that CM Cepharos, the weak exchanger. And if we wanted to bind it tightly, we would use this SP, the strong exchanger. So what we'd have to do is, coming back to the fact you crack the cells open, that might be a pretty acidic pH in the cell, who knows. By the time you've added ammonium sulfate to precipitate things out, you've got a high ionic strength. So what you'd have to do is dialyze your sample into a known ionic strength. So most of you will use sodium chloride to buffer your protein in solution. And so you put in a set amount of sodium chloride that allows your protein to bind the resin, that doesn't compete. Because as you'd imagine, right, if your protein is charged and the resin is charged, salt will only compete. So the concentration you want to use would probably be something like 50, 100 millimolar, whereas when you get up to 300 millimolar and above, then it's going to come off the resin. And the other thing you should think about, right, is cells don't really use sodium at high concentrations. Most cells use potassium. So I wouldn't always try sodium chloride. I'd try potassium too. That might be friendlier to your protein. Off you go. You put your protein in a dialysis bag. Then you'd have a defined composition of salt in your solvent, and it would exchange out. If your buffer is really expensive, then instead of doing one big bulk exchange, you would do 2 small ones, and they'd have the same effect. That matters if you're working on membrane proteins where you've got detergent in your solution, because detergents are massively expensive. So sometimes you do small-scale dialysis rather than large-scale dialysis. You get your protein into a defined salt concentration. Okay, this is a really cool example of a protein. Way back, somebody set a challenge. They said, look, we don't understand how proteins fold. I'm going to make a prize. I think it was almost for $1 million. I can't remember how much. It was certainly a lot of money for the time. And the challenge was, if I give you a sequence that's all alpha or all beta, can you change that, but keep 50% of the sequence the same and totally change the fold? And this is a really good challenge. Normally, right, if things are about 30% the same sequence, they fold the same. So, for example, you know, hemoglobin from humans and hemoglobin from a lower organism, you know, they might have 30% sequence ID, but they both fold to be hemoglobin. So the challenge was here, if you've got an all alpha fold, all helices, can you retain 50% of that sequence but force it to be all beta? And if you could, then we'd understand some of the rules of protein folding. And the first person to do this did this very cleverly. And then they raised the game. They said, all right, can you keep 60% of the sequence, 70%? And it ended up at this, which is amazing.